Good afternoon, students. I'm very happy to welcome you for Zoom class once again. Today we are going to discuss new chapter, that is genetics. And what we are going to say in genetics. Before that, I should define what is genetics. Actually, genetics is a branch of science that deals with the transmission of characters from parents to offspring. That is called genetics, right? Then what we are going to see in the lesson. Let me list out one by one. It is the chapters in which we are going to have following subtopics, right? As I said, genetics, it is a branch of biological science that deals with the transmission of characters from parents to offspring. That is called genetics. The term genetics, it was first introduced by Birds in the year of 1906. He was the man to be introduced to be coined the term genetics, right? As I explained, it is nothing but it is a transmission of characters from parents to offspring, right? That is called genetics. And what we are going to see in the lesson, the first subtopic is heredity and variation, and second topic is Mendelism, and third one is laws of Mendelian inheritance, and fourth one is monohybrid, and fifth one is a dihybrid, and back crosses and test crosses. The next topic is interaction of genes. There it is of following types. They are intragenic interaction and intergenic interaction. And next, one more category: incomplete dominance and lethal genes and epistasis. And 2.6 is a polygenic inheritance, in which, for example, a Pisum certain is given. And pleiotropic genes, we are going to discuss about pleiotropic genes. And next one is the extra chromosomal inheritance. Right? It is up to the cytoplasmic inheritance and a chloroplast inheritance. Right? So this is the outline of this chapter. I repeat it again. Genetics is a branch of biological science that deals with the transmission of characters from parents to offspring. That is called genetics. Then what we are going to say, the first topic is heredity and variation, and the second topic is Mendelism, contribution of Mendelism, history of Mendelism, and works of Mendelism, and success of Mendel in Pisum certiorari. Next, to laws of Mendelian inheritance, law of dominance, law of independent assertment, law of incomplete right. And next, monohybrid, dihybrid crosses, test crosses, back crosses. And 2.5 is the interaction of genes. They are intragenic interaction and the intergenic interaction. And 2.6 is a polygenic inheritance in wheat. And kernel is given, and pleiotropic genes, an extra chromosomal inheritance, and cytoplasmic inheritance. So this is the one second. Just I would like to touch the outline of genetics. First of all, let me take heredity and variation. Heredity It's nothing but it is a transmission of characters from parents to offspring. It is nothing but transmission of characters. Transmission of characters from parents to offspring. From parents to offspring. From parents to offspring. So that is called heredity, right? Heredity is nothing but how these characters are transmitted from parents to offspring. That is called heredity. Before that, I should introduce the types of genetics. As I said, genetics is a branch of science that deals with the transmission of characters from parents to offspring. That is called genetics. Now, this genetics broadly classified into following types. The first category is classical genetics. The first type is classical genetics. And second type is molecular genetics. The third type is population genetics. And fourth category is quantitative genetics. I repeat once again, genetics. It is of several types, but here there are four types are given. The one is classical genetics, and the second type is molecular genetics, and third one is population genetics, and fourth one is quantitative genetics. I repeat once again, genetics, it is further classified into following types. The first type is a classical, the second type is a molecular, and third type is a population, and fourth one is a quantitative genetics, right? So what are the difference between each one, right? First of all, let me take classical genetics. Actually, classical genetics is it is also known as a transmission genetics. Classical genetics is also known as transmission genetics, right? Transmission genetics, because it's a branch of science that deals with the mechanism of transmission of characters from parents to offspring. That is called classical genetics, or it is also known as transmission genetics. The transmission of characters from parents to offspring. That means, how does a character transmit from parents to offspring? I mean, from the parental character, right? How it is a transmitter to next generation through recombination of genes, right? By the fusion of gametes from male plant and female plant, by which the character is a transmitter from parents to offspring. That is dealt by classical genetics. It is also known as a transmission genetics. Second one, molecular genetics. It is also a branch of genetics that deals with the structure and function of genes at molecular level. While transmitting characters from parents to offspring, it is going to deal about the structure and the function of a gene at the molecular level. That is to underline. Please underline. Molecular genetics is a branch of genetics that deals with heredity from parents to offspring. I mean, including the structure and the function of a gene at the molecular level. It is called molecular genetics. And third one, population genetics. A population genetics is nothing but it is a branch of genetics that deals with heredity in a groups of individual for traits which are controlled by only few genes, right? Which are controlled by only few genes. That is called the population genetics. I repeat once again. Now this is a population. I'd like to show you. I'd like to illustrate a diagram to understand. You can. Here, the second diagram that you can understand clearly. I'm talking about population genetics, in which you can say the population genetics deals heredity in a groups of individual for a traits which is going to be determined by only few genes. That means few genes are going to determine particular character in groups of individual. It is a transmitter. It is dealt by a special branch of science that is called population genetics. So what is the right definition of population genetics? Population genetics is nothing. It's a branch of genetics that deals with the heredity. You know very well, I have explained what is heredity. Transmission of characters from parents to offspring, right? Where in a groups of individual for what? For attaining, for getting a traits which is determined by only few genes. That is called a population genetics. And fourth one is a quantitative genetics. Actually, quantitative genetics is nothing. It's a branch of genetics that deals with simply a groups of individual where a character or trait is going to be governed by many genes. That is the word to underline, students. Please remember, it is going to determined by many genes, right? They are the example of quantitative genetics. Quantitative genetics, I repeat once again, it is nothing. It's a branch of genetics that deals heredity in groups of individual where a trait is governed by several genes or many genes are involved in determination of particular traits, while a transmitting character from parents to offspring, that is called quantitative genetics. Well, this is a types of genetics. Now let us continue heredity. As I said, heredity is nothing but a repeatedly I'm saying heredity because we are going to use the term heredity for a number of times. Students, please remember, you can understand again and again. That's why I'm using heredity repeatedly, right? As I said, heredity is nothing but transmission of characters from parents to offspring. How this character is a transmitter? Once again, let me illustrate that you can understand. This is a picture, a flowchart that you see. There are two types of I'd like to compare. I'd like to point out exactly what is heredity. Now, this is a flower in which you can see there are two colors. One is a purple color. The purple color of the flower is determined by a particular gene. The purple color is a parental one. From the parental, the character transmitter from where? So the character is transmitter to F1. I mean, F1 means the first field generation, or it is also known as the offspring. Right? Offsprings are nothing but like a child-like structure, right? Next generation. When we take a purple color and the white color flower or parental, while crossing, this is the parental purple color flower of the plant with the white color flower of the plant, while crossing with each other, F1 was produced as pink color. So this purple color is retained. It is going to be derived from the parental character. So both in parent and offspring, you can find the same color, that is the purple color. The purple color is one of the character. It is a transmitted from parents to offspring. So such a mechanism is known as a heredity. Now you can understand. Heredity is nothing but it is a transmission of characters from parents to offspring. So I have taken two types of parent. They are purple color and white color parent. When we cross with each other, F1 was obtained as a purple color. This character is a transmitter or obtained or derived, produced from the one of the parents which are involved in hybridization technique. Now you can understand what is heredity. Students, I repeat once again. Purple color plant is one of the parent and the white color flower of the plant is also considered to be as another parent, whether male or female parent. So while crossing with each other, F1 was produced with the parental characters. So that is a underline. That is the word to underline, to catch, to understand, right? So the same, the purple color is obtained, produced in F1 generation or offspring. So this is a transmission of character from parents to offspring. It is known as a heredity, right? Now heredity is made possible by combination of genes. I mean, recombination of genes. Here you can say there are two different parents are involved, right? This is a purple color parent and a white color parent. So F1 was produced as a purple, the same color. Again, when we selfing, right? When we do selfing, the F2 will be two different color. The F2 will be two different color. The first one is a purple color. The second one is also purple color. And third one is also purple color. And fourth one is white color. So the ratio, I can say, out of 4 progeny, there will be 3:1. That is a ratio which we obtained by one of the crosses. That is, it may be monohybrid, right? So this ratio, so in these 4 out of 4 progeny, you can find variation. That means variation is nothing but a group of a species or organism belonging to the same categories or same groups of organism, in which, right, you can find differences. That is called a variation. My dear students, please understand. Next we pass on to variation. My dear students, we are discussing about variation. No, variation is nothing. A group of species or organism which are belonging to same category, that shows a difference. That is called a variation. I mean, a group of individual. If you take a forest, or just I'd like to illustrate. Now this is a forest. in which you find a number of species, number of plants, right? All the plants are belonging to same categories, right? In which some plants are showing difference. That shows a difference, right? Some individual shows difference. I mean, it possesses certain characters or different one, right? This is called variation, right? It shows a difference. I repeat once again, what is variation? A group of organism or species which are belonging to the same category that shows a difference. That means some character may indicate difference, right? That is called variation. Now, this variation is caused by recombination of genes which are transmitted from different parents, right? So, as I said previously, just we can see, right? This is an example that you can take it. Here, purple color and white color is there, and F1 will be purple. Again, this is a purple and white. When we mix with each other, F2 will be variety of color, right? So that is called variation. Oh, simply I can say variation is nothing but a group of organism or a group of individual which are belonging to the same categories in plant kingdom that makes a difference, that shows a difference, right? That is called a variation. Now, this variation, it is of two types generally. They are continuous variation and discontinuous variation. My dear students, variation, it is of two types. First type is a continuous variation, and the second category is a discontinuous variation, right? Variation, it is of two. They are continuous. And second category is a discontinuous. Dear students, variation, as I said, it is of several types. First type is a continuous, and the second type is a discontinuous variation. Discontinuous variation. What are the difference between continuous variation and a discontinuous variation? A continuous variation that shows continuous gradation, right? It shows a continuous gradation. Generation after generation without any gap, there is no breakage. An expression, such a character, particular character, right? That is called a continuous variation, right? This continuous variation, it is to be determined by both the environmental factors and the genetical factors. Genetical factors, it is determined by both genetical factors and environmental factors. So it is determined by both genetical factors and the environmental factors. The character of a plant and an organism, it is determined by both the environmental factors, environmental condition, and genetical factors. Right? That is called the continuous variation. One more thing I can say. It is also known as continuous variation. It is also known as quantitative inheritance. It is also called as quantitative inheritance. Quantitative inheritance. That means quantitative inheritance means a transmission of characters which are determined by several genes. Here, I repeat once again, there are many genes are involved. So this determination of characters in an organism by the involvement of several genes, right? So that is the nature of a continuous variation, right? I repeat once again. Continuous variation is nothing more. It is combined by both the genetical factors and the environmental factor. It means continuous variation is caused or it is made by the combination of both the genetical factors and the environmental factors by the fusion of male and female gamete in an organism, right? It is determined by, as I said, both the environmental condition and the genetical factors, and it is also known as quantitative inheritance because it is determined by, the inheritance of particular character in an organism is determined by several genes. Some genes are, obtained from parent, one of the male parent, and some genes are obtained from one of the female parent. So both genes are involved in determination of particular character in case of continuous variation. For that, I can say some example. I would like to give some example, right? A human skin. Human skin and color. Human skin and color. Human color. Human skin and human color or the example of continuous variation. What about a discontinuous variation? Discontinuous variation, it's a limited form of variation. I repeat once again, my dear students, it is very easy to compare and store in your mind. So simply discontinuous variation means it shows it is a limited form. It is a limited form of variation. The variation is not constant. It is going to be expressed in particular generation and next generation it may not be expressed, right? It is called a limited form of variation. Second one. For that I can say some example. Length of the style. Length of style in case of primula. In primula plant, in which you can find length of the style, and one more, height of the tree, height of Pisum sativum or pea plants, right? A pea plant in which height is also determined. In one generation it is going to be expressed, in next generation it is going to be suppressed, right? Due to environmental factors and genetical factors. So these discontinuous variation, it is only determined by only genetical factors. Here genetical factors are only involved, right? Whereas the phenotype ratio of discontinuous variation in an organism, the phenotype ratio, it is not affected. It means phenotype, it is going to be unaffected by environmental factors. I repeat once again, discontinuous variation is nothing but it's a type of variation in which that shows an organism that shows a limited form of variation because it is determined by only few genes. The genetical factors are only responsible for determination of particular character, and the phenotype ratio of an organism is not affected. It is unaffected by the presence of genetical factors in an organism, right? And for that, some example I'd like to give. The one is length of the style in case of primula. If you take a first generation, the style is very long in case of primula. The next generation, it's not going to be expressed in the same height. It is to be suppressed. So the length of the style, this character is not continuously expressed from one generation to another generation while transmitting of characters from parents to offspring. Because it is to be determined by only few genes or this that shows limited forms of variation. because the phenotype ratio is not affected. It is to be unaffected by the presence of genetical factors. So this is about simply the continuous variation, right? This continuous variation, it is also known as the qualitative inheritance. My dear students, please understand, it is also known as qualitative. Qualitative inheritance. Because the quality of an organism is determined by only particular character, is expressed not continuously, only limited form, right? It is in the form of only limited. So this is a difference between continuous and discontinuous. For that continuous variation, I can say some examples are given here. You can see human color and human skin. Whereas discontinuous variation, just I have given some example. The height of bison sativum and the length of style in case of primolum. So there are two examples that I have taken and given for discontinuous variation, right? What is variation and types of variation that we have discussed. Students, please remember, it is very important area. What is variation? All right, short notes on continuous variation. You can expect this question for 3 mark question, right? Write short notes on continuous variation, right? Or which of the following is an example of discontinuous variation for 1 mark question, right? This is a 1 mark question. Or as an example of continuous variation, right? It is going to be asked in your examination, right? Human skin and human color of a human and the skin of human. They are the example of continuous variation, right? Students, hoping that you could understand, right? Or next we pass on to Mendelism. This is very interesting area. Great Johann Mendel is the father of genetics. Great John Mendel, as was born in Austria, is a monk, right? Has become as a father of genetics. Next we pass on to Mendelism. So the contribution of John Mendel to the world, that is called Mendelism, right? Mendelism is nothing but the work and the contribution of John Mendel to the pea plant, right? That is called Mendelism, because he has contributed a lot regarding genetics, regarding transmission of characters in case of Bison sativum. Actually, John Mendel was born on July 22nd in the year of 1822. I'd like to talk about the history of John Mendel. Right? Johann Mendel. Mendel was born on 22nd July, the year of 1822. Actually, who is king? He's an Austrian monk. Basically, he's an Austrian monk. Basically, he's an Austrian monk. I'd like to show you. Was born in a small village. Right? He's a great John Mendel. Actually, he's a father of genetics. He was born in Austria, right? Was born in a small village of Eisendorf. Eisendorf. Right, in Silesia, right? Recently it is known as, right, the name was converted, the name was changed as INC. Actually, he was born in Czechoslovakia, right? Was born in, John Mendel was born in, I'm talking about the history of John Mendel, was born in Czechoslovakia. Was a monk, right? Was a monk. Then after completion of his school education, he went for Vienna. Then he has got a job, that is a teacher post. The rest of his time has been spent in the garden of Mentalism Sadivam. Actually, he had been spent more time in garden. Then he had concentrated more on the study of plants, that is botany and mathematics. And physics. So he had concentrated more and more on the study of plants and the mathematics and physics. And later on, after a few years, he was staying in Vienna, then went to Brunn. Then he published a book, right, in Brunn. In Brunn Society, in which he has concentrated more on the study of Pisum sativum plant, right? P plant. Pisum sativum. Pisum sativa is a binomial of P plant. And finally, he has concluded, he has spent more than 7 years. He started his research work in Pisum sativum right from 1856 to 1863, right? In the duration of 1856 to 1863, he got a lot of experiment work on Pisum sativum. Almost 24,000, not 34 species, right? 24,034 species has been experimented by John Mendel, right? In between 1856 to 1863. So finally, he published a paper in Brunn, right? The Society of Natural. He was a systematic researcher. He was the first systematic researcher and published a book, that is a Plant Hybrids. That is the name of the text. Plant Hybrids. The plant hybrid that describes about hybridization technique. I repeat once again, the findings of John Mendel, which was published into a textbook, that is a Plant Hybrids, which was published in the year of 1866, right? 1866, the paper was published in Bloom Society of Nature, right? So this is simply something about of history of John Mendel, right? I repeat once again, John Mendel is an Austrian monk, was born on 22nd of July in the year of 1822. Was born a small man, that is Eisendorf, Silesia. Recently it is known as the INC. And I had more concentration, more interested in case of botany, maths and physics. and basically he was a monk, and he went to Vienna for higher studies. Then he got a job temporarily and as a teacher, and he continued his profession in Vienna. And after that, he went to Brown Society, and he spent more time in Paisam Satyagam, which was planted in his garden of Brown Society. Then he spent more time on Paisam Satyagam for research work. Then he was finding a number of variations among Paisam Satyagam. So all these variations are collected and published in a book, that is plant hybrids. And this presentation was published in the year of 1866. Hence, he was meant to be published, considered to be as a first systematic researcher in case of hybridization, right? That's why he became as a father of genetics. I repeat once again, this is the way that he had come across in his life, and so he became a father of genetics. John Mendel is a monk, basically, right? So next we pass on to Mendel's success. What are the reasons that he had applied, he had followed? He had taken some interesting character, right? So this is a nature of Bison sativum plant. This plant has been taken by John Mendel to do hybridization technique, and he had concentrated more and more on hybridization techniques and varieties that he has obtained from Bison sativum, because it's an annual plant, right? The name of the plant is nature of the plant is the annual plant, and he conducted a number of experiments, only self-pollination. Yes, concentrated only self-pollination, right? Self-pollination. Hence, it could make a number of findings, number of variation is a Bison sativum, right? That is one of the reasons. And he also maintained accurate data and statistical values, right? He had maintained accurate data. Accurate data and statistical values, right? In case of Bison sativum. And almost all the plants are in which the characters are determined by only. He concluded all the inheritance of character determined by only genes. Instead, it was called by him as a factors instead of genes. No, genes is a functional, structural and functional unit of heredity. I repeat once again, genes are nothing but it is a structural and functional unit of heredity. Instead of genes, these hereditary characters are determined by factors, according to the John Mendel. John Mendel, who concluded that instead of genes, he concentrated factors. He believed that the factors are responsible for carrying a particular character from parents to offspring. So that is a reason, one of the reasons for Mendelian success, right? On talking about the Bison sativum is an annual plant. It is very easy to grass. It is very easy to hybrid, right? It is very easy to concentrate. It is easy to maintain the values and data, etc. Right? So these are all the results that he obtained by John Mendel, right? The experiments are on pea plants. What type of experiments are conducted by John Mendel on pea plant? The first one, it is First of all, I have selected Pisum sativum plant. It is an annual plant. It is an annual. Pisum sativa, it's an annual plant. The second one, it is very easy to pollinate. Hence, as concentrated self-pollination. And the flowers are very large in nature, hence it is very easy for emasculation. It is very easy for emasculation. It is very easy for emasculation, right? And it is very easy to maintain the grow. So these are all the experimental work which were conducted by John Mendel, right? So next he has concentrated. There are 7 contrasting characters. John Mendel was concentrated. There are 7 contrasting characters. What are the 7 contrasting characters? Let me point out. While crossing, he was finding such a characters in between 1856 to the contrasting 7 contrasting character. These characters were found in the year of 1856 to 1863. During this year, John Mendel who found this character. A tall man, I mean, stem, length of the stem, which is determined by capital T, capital tall character, and a dwarf character, that is the first character, right? So this height of the plant is determined by capital T, and small t, right? So this one of the character that shows a contrasting character. One is the height, another is a dwarf in nature. Second one, the pod shape. The shape of the pod is considered by John Mendel. One is the inflated, is the dominant one, and the second one is a constructed. While crossing inflated with the constructed, F1 will be definitely inflated in it. So this is the second contrasting character was taken by John Mendel. Third one, shape of the seed, which are determined by round and yellow, right? So this round color is a dominant one, and wrinkled color is, I think, the shape of the seed. Wrinkled one is a recessive character. So while crossing round shape of the seed and this wrinkled shape of the seed, F1 was produced as a round seed. The next one, yellow color of the seed, which is determined by yellow color is a dominant one, and the green color of the seed is determined by recessive character. So while crossing yellow with green, F1 will be yellow. It is also one of the contrasting character. And flower position, one is the axial condition is a dominant one, and the dominal condition is a recessive character. So while crossing axial with the dominal, F1 will be as the only axial. So this is a 5th character, right? And 6th contrasting character, color of the flower. So one is a purple color is a dominant one, and the white color is a recessiveness. While crossing purple with the white, always F1 will be purple because dominant expresses first and recessive expresses later, or suppresses dominant character sometimes. The next pod color, green color is a dominant one, and the yellow color is a recessive character. So these are all the 7 contrasting characters which were taken by John Mendel, right? Students, so thank you for joining. If you have any questions regarding the topic, you can ask, right? Otherwise, just make an assignment and try to send your respective subject teacher What are the assignment one? First one, take down. The first question. Define genetics. Second one. What is variation? Third question. Write about the types of genetics. The next one. Write short notes on continuous variation. Next, mention the 7 characters taken by great John Mendel. Mention the 7 contrasting characters. Seven contrasting characters. Mendelian. Students, thank you for joining. If there is any question, please ask. Students, am I audible? Thank you. Thank you for joining.